Hi everyone. So this is my 4 millionth time to try and film this for you guys. This is the much awaited chapter 14 PowerPoint. So let's talk a little bit about what the contents of chapter 14 are and where we're going to spend our focus. Chapter 14 is a continuation from our last chapter that's basically going to focus in more on those various classes of RNAs that I talked about during the last lecture series. So if you recall, there were all these different types of RNAs. There were things like mRNAs we talked about. Which those are messenger RNAs, and hopefully you recall that those are the protein encoding RNAs. There were tRNAs, which hopefully you remember are linked to amino acids, and those are going to be important for translation. There were rRNAs, which hopefully you recall those are in the ribosome. Ribosomes are made up of both RNA and protein. And then we had some other classes that you maybe hadn't heard of before, things like microRNAs. We're going to talk some more about those today. And small interfering RNAs. And long non-coding RNAs. So this chapter's focus is discussing basically what happens to these RNA molecules after they're copied from the DNA template. Usually these molecules undergo a series of chemical modifications to make them their mature form, and that's a lot of the mechanism that's discussed in this chapter. Now, I'm going to spend a lot of my time focusing in on these sections here from the chapter, but there are a couple of things that I do want you to know from earlier parts of the chapter. So let's just jump right in. So in talking about what kind of processing occurs to RNAs, the one class of RNA that I'm really interested in you understanding at least a little bit about is messenger RNA. And that's because messenger RNA is often what geneticists are concerned with, since it is what is directly translated into proteins. And of course, proteins are sort of the working unit of the cell. All the cellular physiology is the result of protein action. So that's where we spend a lot of our time thinking in terms of genetics. We think about the protein coding genes. Now, let's think back to what a gene looks like in the DNA form. So I'm just going to draw like a little rectangle. That's going to represent our DNA. And you'll remember that the transcriptional unit, whether you're prokaryotic or eukaryotic, usually it has a promoter region. I'm just going to have it be P. So this is DNA here. Okay. So that's your promoter region. Then you have a coding region. Okay. And then you usually have some sort of a terminator area. I'm going to call that T, where the transcriptional process shuts down. Okay. Now that's the DNA. Let's talk about in prokaryotes what the RNA molecule that's generated looks like, because we didn't really talk a lot about that last lecture. So let's assume that we've gone through transcription and now we have a messenger RNA. And in prokaryotes, we don't have to do a lot of processing to get a mature RNA, because prokaryotes don't have these messy introns and other things that eukaryotes have. But if we could zoom in and look at the resulting RNA molecule, the mRNA molecule, after transcription, you're going to have a 5' end, you're going to have a 3' end, okay? And we're going to divide this into 3. We're going to have the coding sequence in the middle, which weirdly is often abbreviated CDS. That's always bothered me, because CDS doesn't directly correspond to coding sequence, but yep, there it is. Okay. Then you have two, what we call UTR regions, for untranslated region, okay? You're going to have a 5' UTR, and you're going to have a 3' UTR. The 5' and 3' UTR are exactly what they sound like. These are regions of the messenger RNA that they don't correspond to amino acids. They don't get turned into part of the protein. In particular, the function of the 5' UTR is that it has a sequence in it that is complementary to the ribosomal RNA sequence in prokaryotes. And so what do we mean by it being complementary? Well, because it's complementary, let me write down complementary. Because it's complementary, then that means that the sequence here is going to bind to the ribosomal RNA sequence. And so what the 5' UTR area does is it helps the messenger RNA to bind to the ribosome so that translation can then begin. All right. The 3' UTR, along with the 5' UTR, also helps to contribute to the overall stability of this messenger RNA. I've told you before that RNAs are particularly vulnerable to the activity of a set of enzymes called RNases. The reason that those exist is because, you've got this messenger RNA, you've got genes that have been activated, but then the cell's situation might change and you might no longer need to translate and transcribe a particular protein. So you don't necessarily want this message hanging around forever. So there's a little bit of a self-destruct mechanism that's built into this in that the RNAs can chew up these mRNA molecules. But we kind of need a Goldilocks scenario. While we want to chew up these mRNA molecules over time, we don't want to immediately digest them because then it's done us no damn good to transcribe it in the first place. So if a nuclease, or like an RNase, and a nuclease enzyme starts chomping on the end here, then basically you have a little bit of a cushion before you get into the coding sequence of the mRNA. Now, everything I just described for you here applies to prokaryotes, okay? But I want to talk a little bit about what happens in eukaryotes, which is what we have in the figure here. So let me change slides so I've got some room to write. Now, in eukaryotes, we know, of course, that we have a different structure to our DNA. And perhaps the first and biggest difference is that we have these introns, okay, that are in between the exons. And of course, you remember, I always say that exons are excellent. This is the part that we actually use. Introns are non-coding, so this does not correlate with any amino acids, okay? So we got to get rid of these intronic sequences. There's also some other things that happen to an mRNA molecule in eukaryotes in order for it to be considered a mature mRNA. So I'm going to draw us an mRNA just like I did on the last slide using my little rectangle here. Okay, and again, this is going to be the 5' end of this mRNA, and then this will be, of course, the 3' end of this mRNA. So the first thing that's going to happen is we've got transcription here that's occurred, and we have a pre-mRNA molecule right here, and that simply means that we have exons and introns all paired up together. So the first thing that's going to happen to this piece of pre-mRNA is we're going to add, see this little blue region right here? At the 5' end of this molecule, we are going to add what is called a 5' cap, okay? And that cap is basically, what happens is a guanine is added along with 2 methyl groups, and this is the chemical abbreviation for a methyl group, okay? So what that does, so we've chemically modified this cap. This cap, the function of this cap is that it helps the ribosome recognize the mRNA. So it helps in ribo recognition. We're going to learn more about that a little later, okay? So this is going to be a place where we can get translation started, all right? The next thing that I want you to know is that, can you see here, they've got this part that says 5' untranslated region. That was actually transcribed in this first exon. So a mature mRNA, or even a pre-mRNA, is going to have a UTR up here, so an area that does not correspond to the protein. Then you're going to have the coding sequence, CDS. Again, that is a stupid abbreviation. And then you're going to have another 3' UTR here, okay? So there's an area at the end of the molecule that's not translated as well. So far, we've gotten all the way through here. Now, at the very end here, what's going to happen are two things. First of all, at the end of our 3' molecule, we are going to add a series of A's, okay? You may have heard of this before. This is called a poly A tail. All right, and this can be somewhere between 50 to, I think I read somewhere 1,000 A's. That seems like a lot of A's, but you end up with this polyadenylated tail. So what does this poly A tail do? And so you can see they've added the poly A tail here. So we have our cap, 5' untranslated region. We still have the introns, but we've got our polyadenylated tail. Basically, this is another area that's going to help in translation. Proteins are going to bind to the poly A tail. They bind here. This whole thing will form a loop. This protein will loop back around and connect to this, and then it will be recognized by the ribosome. So this is helpful in ribosomal recognition as well. And then again, you know, these UTRs, they just sort of function as a stabilizer for this mRNA molecule. The very last step in mRNA processing is getting rid of these introns so that we have a mature mRNA here with a methylated cap, an untranslated region, a protein coding region that does not include our introns, a 3' untranslated region, and a poly A tail. Okay, so let's look at a quick animation of what that looks like. Let's see here. I'm going to change the screen share. And I want the desktop. I want. There we go. And these animations are all turned on for you to look at, but let's go ahead and watch. They were turned on earlier. I'll make sure they're turned on for you guys. Oop, that's not what I wanted. That's the activity. I want the animation. There we go. Sorry about that. All right.
A eukaryotic gene is composed of upstream sequences such as the promoter, the RNA coding region, and downstream sequences. Many RNA coding regions consist of coding regions called exons and non-coding regions called introns. Many genes also contain a 3' consensus sequence. Before an RNA transcript is translated into protein, its pre-mRNA form is extensively processed. The first processing step is the addition of a cap at the 5' end of the pre-mRNA. Capping takes place rapidly after the initiation of transcription. RNA processing continues with polyadenylation. First, the pre-mRNA must be cleaved shortly after the 3' consensus sequence. Then, from 50 to 250 adenine nucleotides are added to the cleavage site. The next step in RNA processing is the removal of introns. An intron contains 3 consensus sequences critical to splicing: a 5' splice site, a branch point, and a 3' splice site. In the first step of splicing, the 5' end of the intron is simultaneously removed from the upstream exon and attached to the branch point. In the second step, the 5' end of the downstream exon is removed from the intron as the exon is spliced to the upstream exon. Splicing continues until all introns are removed. The mature processed transcript is now called messenger RNA or mRNA and contains the 5' untranslated region, the protein coding region, and the 3' untranslated region. The mature and correctly processed mRNA is transported out of the nucleus and into the cytoplasm, where translation by a ribosome can take place. The failure of any of these steps of pre-mRNA processing has consequences for gene expression. For example,
Okay, I'm going to stop it right there. I just want to go back for a second. So do you see how this is looping over? This is part of the initiation of translation. So you can see that the poly A tail is looping over. That's kind of why it's there. Okay. All right. Back to our PowerPoint. So in thinking about this process of splicing, one of the things that I wanted to point out is that, remember we were talking about different classes of RNAs, and we had small nuclear RNAs, and they combined with proteins to make something called SNRPs, small nuclear ribonuclear protein, SNRPs. These SNRPs, there's multiple different SNRPs. I think there's like 50 of them. And they work together to form what is called the spliceosome. And so all of that business that you saw in the animation where the introns were, looping over on themselves and then getting cut out, that cutting is being done by the spliceosomal apparatus. And what it's using is specific sequences in the introns. There's usually a splice sequence here and a splice sequence there. And these spliceosomes recognize these sequences, and that's how they're identified as introns that need to be cut out of the mRNA. Now, one interesting thing about this whole process is that you don't always have to splice an mRNA the same way. There's something called alternate splicing that often occurs. So many moons ago, we used to, in genetics, think that one gene equaled one mRNA equaled one protein, okay? So it was sort of the one gene, one transcript, one protein hypothesis. What we're finding is that that's not necessarily true. You can have an mRNA that maybe has an exon 1, exon 2, 3, 4. So 1, 2, 3, 4. And depending how you splice and put these back together, maybe you have exon 1, exon 3, and exon 4 in one protein, and maybe you have exon 1, 2, 3, 4 in another, or you can have 2, 3, 4, whatever. You get where I'm going. Depending on how these are spliced and put back together, you can end up with either entirely unique proteins or what we call isoforms of the same protein that maybe have slightly different functions from one another. Let me give you a real world example of that. I'm just going to erase this because all of this is, hello, there we go. Because all of this is kind of explained in this figure from your book. So this figure demonstrates alternate splicing in a particular gene here. And in the thyroid, what happens is this gene gets transcribed, and you can see that the quote unquote mother gene here has 6 different exons interspersed by introns in between all of them, right? So the pre-mRNA includes all 6 of those exons and their associated introns. But here's where alternate splicing occurs, where the spliceosome comes in. If you're in the thyroid, what happens, so we're here on this side, what happens is the spliceosomal apparatus is going to splice out the introns, and it's going to cut everything off right chow, so that your resulting protein is made up of exon 1, exon 2, 3, and 4. And that protein encodes a peptide hormone called calcitonin. Calcitonin, you may have heard of it, it actually regulates calcium and phosphorus levels in the blood. But this same gene, okay, gets transcribed, and in the brain, what happens is the spliceosomal apparatus does not stop after exon 4, it actually splices everything together. Exon 1, 2, 3. Oh, sorry, no, there's no exon 4, is there? Splices almost everything together. No exon 4, then 5 and 6. So you can see that it's using the pieces parts here to put together an entirely new protein. This protein is called calcitonin gene-related peptide. So basically, it's not a great descriptive name, but it's telling you it comes from the same gene as calcitonin, but it's its own thing. And what it is it is a vasodilator in the brain. So a vasodilator is, I spelled dilator wrong, is it just D-I? Dilator, I think I spelled that wrong. Vasodilators basically open up blood vessels, right? So it's a vasodilator. Simultaneously, it is involved in pain reception. Totally different function, totally unique protein derived from the same gene. So you can see that this idea that geneticists had that one gene codes for one protein, we're finding that this is not necessarily true. We're actually finding this more and more every day. You guys know that we can sequence DNA now, and we can sequence RNA. And as our sequencing technology gets better and better, we find these little RNA transcripts that seem to be related to genes, but they're in different forms. We find more and more alternate splices all the time. Okay, so that's kind of what I want you to know about processing of messenger RNA. And I'm not really going to hold you responsible for some of the other stuff in the chapter. There's some stuff on transfer RNAs, et cetera, et cetera. But I am interested in talking about two sections at the end of the chapter. And the first one I want to go over with you guys has to do with the phenomenon that I mentioned briefly in one of our earlier lectures, RNA interference. Okay, interference. Golly, my spelling is so off today. Okay, so let's just kind of think back for a second in talking about the central dogma and how that relates to gene expression. And when I say gene expression, let me be really clear. I mean genes that are turned on or turned off, genes that demonstrate a protein product, or genes that do or that are not doing that. So if you think about the central dogma, it says that DNA is transcribed, of course, to RNA, which is what we have been talking about. And then where we're going next is we're going to take our RNA molecules and we're going to turn those into protein. Again, the working unit of cellular physiology. Okay, so in order to control what goes from here to here, you can imagine that the cell pretty tightly governs that. It's a very energy intensive process. It's a process that's going to change the way the cell functions. So lots and lots of cellular control over which DNA is actively turned into protein. Now, where can we control it? Well, lots of different points. For example, we can control it at the level of the DNA. How might we do that? Well, you guys have seen that we can have things like chromatin modifications, where we're packing histones closely together so that you never ever get that DNA transcribed. You can have things like DNA methylation, which if you have that in the promoter of genes, I'm just going to put CH3 for methylation. If you have DNA methylation in the promoter of genes, you're effectively silencing the transcription of those genes as well. So you can control basically what DNA is accessible to the transcriptional apparatus. You can also exert control at the transcriptional level by controlling the activity of transcription factors. Hopefully you guys remember from our last set of lectures, I think I showed you a slide where we had a steroid hormone that bound to a receptor molecule, and that receptor molecule then traveled to the nucleus where it bound to the promoter of target genes to turn transcription on or off, okay? So hormones and other things can act upon this area right here to turn transcription on or off. But we can also exert some control after we've already gone through all the trouble of transcribing DNA into RNA. We can exert control here at this level, at the RNA molecule level. Let's say you've already made it, but for whatever reason, the cell does not want this to get translated into protein. And that is kind of the realm of RNA interference, okay? RNA interference is going to act to break down these RNA molecules to make sure that they don't turn into protein. That probably seems kind of counterintuitive. Why would you go through all the trouble of expressing all of this up to here and then breaking it down here? But we actually believe this is a holdover mechanism that originated as a defense mechanism against viral DNAs and now has been co-opted as a mechanism to further control gene expression. So let's talk about RNA interference. First of all, how do we even discover this? It's not been around for that long. Well, no, let me take that back. It's been around for that long. We've not known about it for that long. As a matter of fact, I can remember one of the first questions I ever asked my PhD advisor was, because I went to school and got my master's degree, and then I taught high school for a long time, and I went back to get my PhD fairly late in life. And one of the things that had changed since I had been in graduate school the first time was this understanding of RNA interference. And one of the first questions that I asked my advisor was, what is RNA interference? And can you describe it to me? So basically what had happened was there was a group that was trying to knock down a protein, trying to knock down gene expression to understand what that protein did. If you turn a protein off in a cell and then you see what happens to the cell, that gives you a great idea of what a protein does. And the way they were doing that was they were injecting RNA that matched the messenger RNA into the cell. And they got some knockdown of translation. But they found that if they injected double-stranded RNA, they really knocked down translation. And they couldn't figure out why that was. So the first thing I want you to know about RNA interference is that it deals with double-stranded RNA. Weird, right? We're going to come back to that, so just put a pin in that. All right, so RNA interference limits the invasion of foreign genes, I've got underlined for you here, and it censors the expression of a cell's own genes, okay? So if you had like an RNA virus or something like that, then RNA interference could take care of that viral RNA, but it can also shut down some of your own RNA, all right? So there are two types of small RNAs that we're going to talk about that get involved in RNA interference. One are called microRNAs, which we talked about before. And the second are small interfering RNAs. Okay, so let's talk first about microRNAs. What are they? How do they arise? Well, basically, you have in your DNA codes for these microRNAs. And you can see that we have an inverted repeat here. And hopefully you've seen this a couple times now. When you have an inverted repeat and that gets transcribed into RNA, what happens is the inverted repeats line up. They're complementary to one another and they form a hairpin, okay? So this is a primary mRNA or a pri-mRNA at this point. Then what happens is these tails get cut off and you've got this hairpin structure. And then along comes an enzyme called dicer, which is awesome because it slices and dices this RNA. It cuts off the loop here. See how it cut off the loop? And what you're left with is this double-stranded RNA. And it's short. It's somewhere around 22 nucleotides. Okay, so it's little. When you have these double-stranded RNAs, they get recognized by a protein complex called the RNA-induced silencing complex, or RISC. So RISC comes along, it's like, hey, I see this this is like a flag for this protein. It comes along, and what it does is it binds to it, but it only binds to one of the two strands. The two strands serve as sort of an identifier for RISC, but RISC only uses one of those strands. Then what RISC does is it sort of travels around and it tries to match this sequence with sequences in mRNAs that are floating around in the cytosol. And when it finds a match, what's going to happen is it's going to come along and it's going to cut this mRNA. Nope, sorry, not in this case. It does not cut it here. It cuts it here. It's not cutting it here. It just inhibits transcription. Sorry. So it binds here. And notice how it's binding imperfectly. See how you have a little hump here? It's binding imperfectly. And it's just going to park there. And it's basically going to block translation of this mRNA. Okay. Let me show you a quick animation just to make sure that that's really clear for you guys. So quick animation here. We're going to go back here. Oops. Sorry, my computer's kind of going crazy. All right. That is the one that I want. And I want RNA interference. There you go.
RNA interference in eukaryotes is a potent molecular mechanism that limits invasive viruses and transposons and silences some cellular genes. It is activated by double-stranded RNA. Let's see how cells regulate gene expression using RNAi. Typically, microRNAs produced by one gene regulate the expression of a different target gene. How does this work? As transcription of a primary microRNA gene moves through an inverted repeat DNA sequence, the RNA product begins to fold. Complementary RNA sequences base pair to form a stable, extended hairpin structure with a double-stranded stem. In time, a processing nuclease cuts off the RNA hairpin. Then another nuclease called dicer binds the free hairpin and cuts out a 21 to 23 base pair microRNA. The double-stranded microRNA is then bound by the RNA-induced silencing complex, RISC, which releases one strand of RNA. RISC uses the microRNA to locate a target messenger RNA by base pairing. However, the microRNA does not base pair perfectly with the target mRNA. This imperfect complex interferes with translation of target mRNA, leading to less gene product when compared with normal, unaltered translation. In contrast to microRNA, small interfering RNA base pairs perfectly to target mRNA, with the outcome that target mRNA is cut and eventually degraded.
Okay, so hopefully that kind of helps. Let me go back to our original presentation. Okay, so the other thing I wanted to talk about here is small interfering RNAs. They were mentioned at the end of that video. Right here. Okay, so small interfering RNAs are similar to microRNAs. MicroRNAs actually have their own little genes, okay, and they're being transcribed. These arise from multiple different mechanisms. So it turns out that, let's see here. Many small interfering RNAs are produced from a single RNA duplex usually. So you have this big single RNA duplex, and then what happens is dicer comes along and we don't have a hairpin. Instead, we have this RNA strand, this double-stranded RNA that gets chopped up, sliced and diced to produce these individual small interfering RNAs. They are similar in size. They're also somewhere in the ballpark of 22 nucleotides long. Again, these are double-stranded RNA molecules, and these double-stranded RNA molecules are recognized by this RNA-induced silencing complex, or RISC. So RISC is going to recognize this. It's going to bind to one of those two strands, and then it's going to go around and it's going to try to match this strand with existing mRNA. Now, here's where it differs, as it mentions in the video. You have imperfect base pairing here. That says just kind of shut down translation, turn it down a lot maybe, and you may still have a little bit of translation that occurs, but the mRNA transcript doesn't get cut up. Here, you actually are going to end up cutting and degrading this mRNA transcript, okay? This is particularly what happens when we have viral RNA. We're going to learn a little later about things called transposons, which are pieces of your DNA that can copy and paste themselves in different regions of the genome, okay? So make sure that you understand about microRNAs and small interfering RNAs. That's one of the things I really want you to get from this chapter. Another thing that I wanted you to get from this chapter has to do with CRISPR RNAs, okay? Well, you're going to have an entire lab on that. And it kind of worked out beautifully that that's what we're talking about in this chapter. Now, the best way to understand and think about CRISPR RNAs, I think, is to think of them as a bacterial immune system. So CRISPR RNAs are found in bacteria usually, and they function to defend the bacteria against viral DNA and plasmids. And here's what happens. So you guys know that viruses work by injecting their genetic material into target cells. So bacteriophages are viruses that specialize in infecting bacteria. So you get some phage DNA in here, and basically what's going to happen is the bacteria is going to take a little section of this phage DNA and it's going to tuck it into its own DNA. And it's done this here. We're going to assume that each of these colors is from a different bacteriophage that this bacterium has encountered, and it's taken just a little sample of that viral DNA and it's incorporated it into its DNA, and in between, it has flanked it with these spacers, okay? So these spacers are clustered, repeated, interspersed, palindromic, something. I always have to look this up. They're going to tell us in a second in the video, but that's where the name CRISPR comes from, are these clustered palindromic sequences here. And so what they're doing is basically flanking this and saying, this is foreign DNA, this is foreign DNA, this is foreign DNA, et cetera, right? So this kind of serves as a bacterial memory. Just like you have immune cells that remember antigens that you've been exposed to. What happens is the bacteria transcribes this array. Now we have a pre-CRISPR RNA. Then you're going to have cleavage by what are called CRISPR-associated proteins, and there's a bunch of them. They come along and they chop this array up at the palindromic sequences, at these sequences here, okay? Then they tuck these various sequences into another Cas protein, and now it becomes what we call an effector complex. And similar to the mechanism of RNA interference, what happens is this complex is going to go along and it's going to look at DNA that's floating around in the bacterial cell, and it's going to try to match this DNA to any DNA that's in the cell. And when it finds some, it knows, hey, that's foreign, we got to cut it apart. So this entire system is basically, it evolved in prokaryotes to act as a defense mechanism against foreign DNA. Now, you have probably heard about CRISPR as it pertains to gene therapy. What we have been able to figure out is that we can engineer different sequences. Okay, we can make up we can make up CRISPR RNA that corresponds to a particular gene that we want to edit. We can then tuck that RNA into a CRISPR-associated protein. We can target the DNA of interest, cut it apart, and insert extra DNA in there, or repair faulty DNA. So we can take advantage of this system to do some gene editing and gene therapy. And that's why you're hearing a lot about CRISPR right now. That's exactly the area of research that people are engaging in, is how to use these CRISPR proteins, well, CRISPR RNAs and CRISPR associated proteins, to edit DNA. So let's look at a quick video explaining that. And then you'll learn everything you ever wanted to know but were afraid to ask during your lab. Oh,
CRISPR stands for...
I should totally change the screen share. Hang on. And I'm trying to find the right screen. Desktop 2, that'll work for me. No, that's it. Okay.
Clustered, regularly interspaced, short palindromic repeats. Discovered in bacterial and archaeal genomes, the short palindromic repetitions of base sequences are followed by short segments of unique spacer sequences. The spacers originate from previous exposures to bacteriophages or plasmids. The CRISPR region is transcribed into a single long CRISPR precursor RNA. Once transcribed, the long CRISPR precursor RNA is cleaved by CRISPR-associated proteins, or Cas, which function as nucleases. Each of the cleaved CRISPR RNAs contains a spacer sequence homologous to a foreign DNA and flanking palindromic sequences. The CRISPR RNA binds to Cas proteins to form an effector complex. In this complex, the CRISPR RNA will serve as a guide RNA to seek homologous DNA sequences in the cell. Upon invasion of additional foreign DNA from the same source as in the CRISPR RNA, the CRISPR RNA pairs with the matching foreign DNA by complementary base pairing. The Cas protein then cleaves the invader DNA by a mechanism similar to RNA interference. In this way, CRISPR RNA serves as an adaptive RNA defense system against foreign invaders for prokaryotes. CRISPR has many potential applications, including for use in genome editing for gene therapy.
All right. So let me go back. Okay. The last thing I want to talk about is this is from section 14.6. And I'm not going to say a lot about this, but I did want to mention long non-coding RNAs. I mentioned them briefly during our chapter 13 lecture set. This is an active area of research. If you're interested in genetic research at all, this would be a place for you to go, because it is hot research-wise, because we just flat don't understand it. Here's what we do know. In eukaryotes, only 1%, if you were to go through and just like look at how much of our DNA is actually coding for proteins, it's only like 1% of our DNA. The rest of it is like introns and other stuff like that, junk DNA, but a lot of it is associated with these long non-coding RNAs, because 80% of our genome ends up getting transcribed into RNA. So long non-coding RNAs are simply what they say. They're long RNAs that do not code for proteins, and they seem to have a variety of functions. We can't seem to find like a singular function that describes all long non-coding RNAs. For example, here's some that we have figured out. So we know that some of them regulate transcription, and here's an example. So there is a long non-coding RNA. This is actually called LINC, for long intergenic non-coding RNA. This particular one interacts with a protein called P53. Now, the P53 protein, you may have heard about if you've heard a lot about cancer or things like that. It is a transcription factor that turns on a lot of genes that are related to apoptosis, okay? But it turns out that lincRNA P21 represses the transcription of this. It basically turns P53 off, and in so doing, it can regulate the rate of apoptosis. So that's cool. Here's another long non-coding RNA we know something about, and this is probably the best studied long non-coding RNA. There is a long non-coding RNA called EXIST, and this X comes from the X chromosome. Basically, recall that you have this whole phenomenon of X inactivation through mechanisms that we still don't understand. So you've got two X chromosomes, right, in the females, and early in development, somewhere around the 100 cell stage in humans. Don't ask me how I know that. That's a random piece of knowledge. What happens is one of these two, one comes from mom, right? One comes from dad. It's randomly decided by the cell through mechanisms we don't understand. One of them starts transcribing a whole bunch of this EXIST long non-coding RNA, just a bunch. And what happens is this EXIST starts coating, like not coding, coating. Like you're coating a piece of chicken, right? It starts coating the outside of the X chromosome, and that serves as a flag for histone modifying proteins and DNA methylating enzymes to come and do their thing to this particular X chromosome and condense the chromatin. Okay? This is going to condense the chromatin here. So this exists long non-coding RNA. Its job is basically involved in epigenetic shutting down of the extra X chromosome and the dosage compensation mechanism. So these are two very different functions, and there are other long non-coding RNAs we found that have other functions as well. So I just wanted you to be aware that they exist. There's many of them. I'd like it if you knew these two examples, but the big take-home message is that we really don't understand them yet. Okay, so that's everything that I want you to know from chapter 14, and I will have your chapter 15 lectures uploaded for you shortly. Talk to you soon. Bye-bye.